Hello, my name is Mr. Chipman. I teach biology and AP biology at Murray High School in Murray, Kentucky. This is Unit 2 Cells 2.5 Membrane Transport. Lots of big ideas in this particular section that are sort of going to permeate, there's a fun word, permeate the rest of the unit. And so I think it's very important to kind of get this basic vocabulary down. So let's do that. All right, so we're going to look at this idea of solute and water balance. First of all, when we're dealing with a solution, right, we have two parts, a solvent, a solute. The solute is the thing that has been dissolved. The solution is the dissolving substance, like water usually is a solvent. If you make salt water, salt is the solute, right? And so in this particular picture, you have a lot of solute on one side of the picture and very little solute on the other side of the picture, right? And so we understand this basic concept of concentration without even giving it a definition. We can look at the left side of this picture and see that it is more concentrated with solute than the right side. This is what we call a concentration gradient, right? A concentration gradient is when one side has a higher concentration than another side, or one area, or whatever, right? If you spray perfume into the center of the room, there's a gradient of that perfume. Where you just sprayed it is going to be very concentrated, and the rest of the room is going to be less concentrated, but eventually you're going to smell it throughout the room because those particles are going to sort of diffuse or move or permeate or whatever you want to say throughout the rest of the room, thus sort of equalizing that concentration, right? That gradient is going to get less and less over time. And so that is the idea that we're going to be looking at. Cells use this sort of thing all the time for homeostasis, for signaling, for energy conversion. There's a lot of reasons that cells use concentration gradients. And so that concept is central to what we're getting ready to learn. And one misunderstanding that my students often have is they will see the concentration gradient as the action that takes place. The gradient is the cause of the action. The gradient is the thing that exists, and the movement of particles is the action, and there are several different names for the movement of particles depending on what's going on. So let's look at those. First one is passive transport. Passive transport requires no energy. Alluded a little bit to this idea of transport in the last video, 2.4, and but here we're going to really deal with it. So with passive transport, there is no input of energy necessary, right? The illustration of the perfume going throughout the room just a few seconds ago required no energy, right? No one was pumping that perfume throughout the room or anything. It was just sort of diffusing naturally throughout the room. And so passive transport is transport that requires no energy because materials or molecules or whatever you're dealing with is going to move this way from a high concentration to a low concentration naturally. No energy is going to be necessary. There's going to be a couple of types of passive transport that we're going to look at. Here, first we have simple diffusion, and then we have facilitated diffusion. Osmosis is next. We'll get there. Osmosis is a little more complicated. And so with simple diffusion, you just have areas moving across a membrane, typically some sort of selectively permeable membrane, because that's what we're going to be using a lot in this class, from an area of high concentration to an area of low concentration. Diffusion doesn't necessarily require a membrane, right? We're going to look at some examples of that, but we usually are talking about a membrane when we're talking about passive transport because we're talking about transport materials in and out of a cell, right? Facilitated diffusion is when that transport still occurs. It's still diffusion in the sense that it doesn't require any energy, right? It's passive transport, but it needs help. It needs facilitation, big word for help. And so it needs some sort of protein channel, some sort of carrier protein. We're still going high to low, right? And so one misunderstanding that often happens here is that when you're talking about protein channels, that it doesn't matter what it is. It's called facilitated diffusion. No, it's the diffusion part that makes it passive, right? And so make sure that you hang on to that. Osmosis is a type of diffusion in that it is the diffusion of water. the diffusion of water. And we were going to talk about this at length in 2.7, and so I'm not going to borrow a whole lot from that, but water is a material, just like a solute is a material, and water will move independently of the solute, right? Because water is not chemically combined with a solute, it's just a physical mixture, and so those two things will move independent of one another, and it causes some interesting phenomena that we're going to look at specifically in 2.7. But make sure that you attach osmosis to diffusion and diffusion to passive transport. This is transport that requires no energy. Active transport, on the other hand, does require energy, right? It's an active thing. It's actively taking place. Energy in this case is typically ATP, right? So there's going to be some exceptions to that, but ATP is the energy source of the cell, or the energy currency of the cell, and this allows materials to go from an area of low concentration to high concentration, right? This is you putting air in your bicycle tire when it's flat. You don't just hook the bicycle pump up to your tire and then sit there and look at it, right? Nothing would happen. Actually, your tire would get more flat because of the passive transport of air out of that hole that you've created, right? But instead, it requires activity. It requires an active input of your energy in order to pump air into that tire, which represents a higher concentration of air, right? Active transport is usually used to create a concentration gradient, right? If you're moving things from high to low, you are creating a gradient. Why would you want to create a gradient? Well, usually to do some sort of cellular work. All of Unit 3, Unit 3, is, sometimes I get my units mixed up. All of Unit 3 is about creating a gradient, right? And how gradients are created using energy of some sort. And so that is active transport. Let's look at bulk transport. It is exactly what it sounds like. This has to do with the transport of large materials. This is also active transport, so you could put this under active transport if you want to do that. Active transport or bulk transport has to do with things like endocytosis. Endocytosis, endo means inside cell, or cyte means cell, so literally bringing things into the cell. This picture is one of my favorites because you have all three different types here. You have phagocytosis, which is the ingestion of solid particles. So here this cell is wanting to eat some sort of bean or something, right? And notice what the cell does. It sort of like reaches out and like pinches off a little piece of itself and creates this little vacuole, or sometimes you'll see it called a vesicle, which is like a temporary storage thing, right? Either way, whatever, right? And it pinches it off, and this is now surrounded by a phospholipid bilayer, which is all the membranes of the cell, right? This should make sense to us. Penocytosis is just the same idea, but bringing in fluid instead, bulk fluid, bulk solid materials. Receptor-mediated endocytosis needs a receptor, right? It will bring things in if those things are able to connect to a receptor. In this particular case, we're bringing just stars in, sorry squares, not today, and when the stars connect to these receptors, it triggers endocytosis. Again, bulk transport is a type of active transport. The opposite of endocytosis is exocytosis. Love this picture because it shows you this phospholipid bilayer. Right here, this cell is trying to get rid of green dots. And to get rid of green dots, it's going to push this vesicle to the extracellular. No, that's the outside of the cell. This is the plasma membrane, right? It's going to push that vesicle to the plasma membrane. That vesicle membrane is going to fuse with the plasma membrane and push those materials out. We've talked about this already, right? We talked about the endomembrane system, the idea of proteins being made in the rough ER, then pushed to the Golgi, and then pushed out of the cell. This is the pushed out of the cell part, exocytosis. A couple of examples of this. On the left here, you have a proton pump. Anytime you see the word pump, right, it's like your bicycle pump. It requires energy. This is a proton pump, so guess what it pumps? It pumps protons. Right, we're going to talk about proton pumps in Unit 3. See them a lot, but it requires the input of energy. Over here on the right, you have this is a nerve cell, and this is an axon terminal, and these are neurotransmitters. And this neurotransmitter, I mentioned earlier that sometimes you'll see diffusion when it doesn't involve a membrane. So a couple things going on here. First, you have exocytosis, right? You have this material being pushed out into this membrane. And then these neurotransmitters will actually diffuse across this synapse, across this space, go from an area of high concentration to an area of low concentration in order to get to the postsynaptic dendrite. Dendrites and all that stuff, words you don't have to know, but diffusion, exocytosis, definitely have to know. I love the token mitochondria in there. Perfect. Let's talk about practice. In intestinal epithelial cells, glucose often enters the cell, even when its concentration is higher inside than outside. Which the following best explains how this occurs? Pause the video and we'll talk about it. Okay, so if we're going from an area of low to high, right, enters the cell even when the concentration of the inside is higher. We're pushing materials into the cell, low concentration to high concentration. How would that occur? It's active transport, right? Let's go find that ABCD. Glucose is nonpolar. Who cares if it's nonpolar? None of this has anything to do, and it's not even true. Facilitated diffusion moves glucose into the cell, right? This is not facilitated diffusion because we're not diffusing. Diffusion is high to low. Active transport couples the movement of sodium down to its gradient to power glucose uptake against the gradient. Love it. I don't even really have to know what any of this sodium gradient uptake, all that. Who knows? I know this is active transport. D, just why would glucose go through aquaporins? It's not even welcome. C is the correct answer. We like C. Hopefully this was helpful. 2.6 is coming soon.